Hi, this is Hailun. Today, I want to introduce the GenomeBiomarker system to show some new view and integration of resequencing products. And as you know, BGI Tech as a regular and transnational high-tech corporation, but didn't have any product or series of products with registered trademark. I think it's abnormal. And now we just want to take the resequencing products as a demo to build up a product series which specialized in molecular marker development with our own trademark. And I want to introduce you this integrated system and some new views on breeding and evolution research. Firstly, I want to show the background of molecular markers. In the late 1980s, researchers began identifying specific regions of DNA that influenced the phenotypic traits in certain species. The efforts soon turned to development of accurate, cost-effective genetic tests that could characterize the genotype of this region in the sample from RFLP, which only using enzyme digestion, to RAPD and AFLP, which involved PCR method. And then short tandem repeats, or we call it SSR, were ultimately replaced with single nucleotide polymorphisms as the chosen marker for genotyping studies. This because SMP present in high abundance within genomes. Also, when screen the high densities for a given species, they enable the efficient tracking of the transfer of genetic regions from parent to offspring. So, we believe the trend should be resolve this problem. How to scale up to large targets with full utilizing the sequencing capacity offered by next-generation sequencing platforms in a cost-effective way. The rapid evolution of sequencing technologies is driving the development of dynamic, scalable, and efficient and low-cost methods of marker development solutions. Our genome biomarker system is an integration platform that enables the swift deployment of NGS-based methods applying to marker development and genotyping and linkage and genetic mapping, population evolution, and conservation biology research of agricultural and aquaculture. We just distinguish two types of markers. One is nucleotide sequence polymorphism, such as SMP, indel, and SV. And one is sequences length polymorphisms and other kinds of markers, such as SSR, RFLP, and also some results we get it from biochemical test. For the first kind of markers consists of more than 7 products using for marker initial development to known loci genotyping. There are some conventional products such as resequencing, target region capture, restriction method, restriction enzyme method consist of our products such like RAD and GBS. And there are also some demo products in this list, such as the capture gene typing and map-based cloning. And some downstream products are taken into account of this system, such as OFISH, which stand for oligonucleotide effervescence in situ hybridization. Yeah, and it's an improved FISH method developed by Agilent. And GMO, which stand for genetically modified organisms, are also involved in our downstream products, and I will introduce them later. And that's the distribution of different products between the cost and marker density across the genome. So we could say PCR-based method is used for hundreds to thousands of base pairs detection. And array-based gene typing and capture-based gene typing, they are used to detect range from kilo base pair to mega base pair. And map-based cloning method is uniquely used to capture region from 10 kilo base pair to hundreds of kilo base pair. And target region capture methods is useful for a fragment whose length distribution from hundreds of kilo base pair to dozens of mega base pair. And also RD of restriction enzyme method could detect thousands to hundreds of kiloloci, which of GBS is about thousands of loci. Resequencing is the most powerful method to generate information of the whole genome, but it's limited to the species which only with reference genome. I will introduce something about resequencing. There is the application of whole genome sequencing. By this method, it's still the most powerful method, especially for complex crop QTL mapping or high-density markers development, such as the hexaploid wheat breeding. And in some other areas, such as gene function evolution research, it's still powerful than many other methods. But there is also problems of WGS. The, mar- the and nowadays we just want to clarify the market requirements and the competitiveness of our bioinformatics analysis. Just because in the first quarter of this year, the projects without bioinformatics analysis are more than 70%. So we just think how can we to sell more bioinformatics analysis of resequencing. I think one method maybe we could do a sales promotion, but maybe it's not useful for the European and also America. So I think one important step we could generate is that we could provide as personalized as ties service for our customer. I think our customer don't need to lose their waste to fit our standard or advanced bioinformatics analysis. We could construct a service to fit their love handles. And this need more works of marketing, project operation, and also bioinformatics updata. And if we had more articles with leading analysis items, I think it should be another advantage of our bioinformatics analysis of resequencing. That's the pipeline of resequencing. I won't show the detail. And in our products, we have individual genome resequencing and also population genome resequencing. So that's the detail. And okay, and that's it for the resequencing. And now I want to introduce something about restriction enzyme methods. Um, in our plan for this product, we just want to put four products in this method. One is traditional RD-seq, and one is RD-seq with assembly. and double digest RAD and GBS. And this protocol, initially established for crops like maize and wheat, have advantages in cost per sample and application in species where there is no knowledge of the genome. The application of this method is especially powerful in mapping populations or closely related groups of samples, such as candidates of genomic selection. If populations are more divergent than expected or targeted, maybe we could to finish an tunable protocol for our project design. And for the double digest RAD, I think we need more tunable number of regions with lower cost than RAD. Okay. 2010, Science has just published the areas to watch of 2011, and one of them showed that RAD tag sequencing may be useful for the adaptation genes research area. Most organisms saturate large numbers of DNA sequences polymorphism that disrupt restriction sites, which allows RAD or GPS tags to serve as genetic marker spread at a high density throughout the genome. That's the first feasibility of these restriction enzyme methods. And RAD stands for restriction site associated DNA. They are a genome-wide representation of every site of a particular restriction enzyme by short DNA tags. And GBS stands for gene typing by sequencing. The key components of this system are we reduced the sample handling and fewer PCR and purification steps, and no size fractionation and inexpensive barcoding. That's the advantage of restriction enzyme methods. We could do it on a simplified genome and with high density and homogeneity, and it's cost effective comparing with maybe resequencing, and it's independent of reference genome. And there is one article published on Nature Reviews Genetics 2011. They cite that a multiplex sequencing protocol designed to provide maximum data at minimum cost. Yeah, they mean the restriction enzyme methods. That's the application of this series of products. And that's the pipeline of RD-seq. We could say we just add adapter to fragments after digestion. And then we share the DNA and to purify RD tags and then get many labeled tags and then sequencing. That's the difference between RAD and GBS. We could say GBS don't have the random share, site select, and the ligation of Y adapters. There is one product that we are recommended for many projects recently. We just want to use the parent reads that we generated. The read one with enzyme recognition size, and then 400 base pair contigs constructed by read 2. So that's the pipeline of this method. So the key point of this method is that we share the DNA sequences randomly, and then we select the fragment size from maybe 300 to 650. So we get the fragments with different length, but they could cluster to the same loci of the genome. So the sequence of read 2 comes from different length of fragment. So we could assemble this part of read 2. And there is another products now we try to do some upgrading on it. Um, about the traditional RAD, we use maybe one enzyme to digest the whole genome, and then we get all of the tags that with the enzyme site. But if we add one enzyme into this system, we just digest the whole genome twice, and then we could get fewer fragments from this step. And so it becomes more tunable than traditional RD. That's the advantage of this method. We could get high SMP accuracy just because the greater read depth per site allows accuracy SMP calling and low data missing rate. Via tunable digestion protocol, we balance size, numbers, and supported reads per site to reduce missing data rate. And it's cost effective than the traditional RD protocol. And also is flexible design because both informatics simulation and wide experiment simulation will be implemented while project designing. And here we show the enzyme we used for these products. And there is the bioinformatics protocol. And for RD-seq and also GBS, we have an improved SMP calling protocol, which we call it DISR. And when we get the reads cluster, then we do one step of error correct. And then we could get high depth region filter. And yeah, maybe you could to pronounce this protocol to our customer because we think it may be some advantage of our RED or GBS. And there is also some problems of restriction enzyme methods, and also is the direction that our updates will focus on. First is the data bias. Just because if we just because neither RED or GBS, we are pulling many samples in one line. And if we didn't did a good to test the quality of the DNA, so the data generated from one line will be biased. And now we are trying to make some upgrade of this problem just by we add one step of quantify of the whole genome DNA. One step is before the enzyme digestion and one step after the digestion. So we could to resolve this problem. And this second problems may be the flexible design because as we know, the biggest corporation of RED is located at USA, the corporation name is Genetics, and also Cornell University, located also at USA, they also provide the RAD and also GBS services. In their service type, they could provide more flexible design for customers' project. So that's one problem that we are resolving now. And also the cost. Maybe many of you know that um, such as Cornell University, they could provide GBS for maybe $30 to $40 for a sample, but the price of our of BGI's GBS is still maybe 50 to 60. So I think in our department of production of the wide experiment, we also have some space to reduce the cost of all of these products. And also, we have to have some promotion of our capacity of sample pooling, just because, for example, Cornell University, they could pooling more than 384 samples in one lab, but we just can pooling 192 samples in one lab. And also recently we have some negotiation with the corporation Genetics, and they are so interested in have some cooperation with BGI. And we are now just finishing our plan and to a discussion on this. And there is the target region capture. And I think it's ideal for species with a priori knowledge for regions of interest and powerful for SMP discovery and fine mapping of the recombination breakpoints. And someone just cites that this promises to be the next wave in NGS-based gene typing. That's the traditional pipeline of target region capture. And I want to talk about some problems of this product. In the past about 2 years, many projects are denied just because they have less sample or the customer just wants to capture a small region. And also the higher cost of this product is also a big problem. And I think we finally we want to build up our own products. And I think maybe some update may resolve this problem. One is we could to build our self-production capture chip, not protected by Angeline or protected by Nipson. Just we BGI, we product the capture chip. So we could get more flexible and cheaper chip to do the target region capture. And also we could update the minimum order of request of the chip, and also update and reduce the cost of library construction. And maybe some database construction and products for important species. Just because we have many data of many important species genome, and we could just to build our database based on this genome, and then we could to push our product that we could do and maybe uh, 20 megabits of sheep genome or uh, 10 gigabits tackle region capture for maybe Panda. Yeah, I think that's the advantage of BGI, and now we could to catch the trend and to do some update of this product. And then it's the capture gene typing. It is similar with target region capture, just because we also use the uh, Angelin or Nibgen capture region capture, but there is some difference between that. Uh, this product, the capture gene typing, could get many points throughout all of the genome. Yeah, it's one of the hybridization-based method and use oligonucleotide specificity to bind to and to isolate complementary sequences. And its cost could be lower than array. And also there will be more benefits for BGI just because we buy all the capture buy all the array from Illumina, and now we are have some negotiation with F Matrix. But anyway, if we can do the genotyping, the vast amount sample genotyping by the capture genotyping instead of microarray based, I think it could provide more benefits for BGI. And it could provide more kinds of variation, not just SMP. For this product, there is still some problem on it. One is the capture specific, and the second is coverage rate, and then is the vast sample polling strategy and stability. And we just have a demo project in our research center, and we have the for the SMP selection, we select the SMP with minor allele frequency more than 0.2, and we just get 120 base pair specific sequences of every loci, every site. And then the sequence should be the GC content should be larger than 35 and lower than 60%. And we finished three kinds of polling strategy. One is a 5 kilo loci and 48 samples polling, and also 5,000 loci and 96 samples polling, and 50,000 loci, 48 samples polling, and use the Nibgen tag region capture chip. Here we show the capture rate and also the coverage rate of this rice demo case. And we could say all of these three strategies get a high coverage rate, and also the capture rate is also so high. So that's the coverage rate distribution of these three strategies. And there is the SSR loci development. In the past 2 years, we have two methods to get SSR loci. One is NGS-based SSR loci development, and one is transcriptome. And now we are evaluating the 454-based SSR loci development protocol. And maybe I will show more information via email. And there is the our improved fish product, and this product is provided by Angelin. and it could be used in the verify genes in a cytobiology way, and maybe we could to put it in our downstream products of our genome biomarker system. Yeah, thanks for your attention. I just put one of my photo on this slide, and this system and many updates are still in an early form, so we really need your suggestions on them. And at last, welcome any questions via email. Thank you.